So today we're going to go back to our scheme of molecular phylogenetic ecology and do the backwards part of it. So we get to ribosomal sequences and phylotypes. Now we're going to talk about using fluorescent in situ hybridization to go back and look at the organisms and their environment. Now we've done this a couple of times already with basic fluorescent in situ hybridization. And so the trick here, of course, is you take the cells, you fix them in a formaldehyde or glutaraldehyde base. This kills them, so we're not looking at living cells. And then you make a probe specific to particular ribosomal sequences, fluorescently label it, soak it in, and you light up your cells and look at them under a fluorescent microscope. This is all pretty basic stuff. There's some tricks to it, and we're going to use a very elaborate version of that in the paper today. Why target ribosomal RNA for that, though? Well, now, from the background of what we're doing in this class, it's pretty obvious because that's the sequence you have. But it turns out that ribosomal RNAs are really, really good for this for a very simple reason. You can target any gene in the genome for this. But if, for example, you target the gene for glyceraldehyde phosphate dehydrogenase, how many targets are there going to be in a cell? One, right? And so you put one fluorescent molecule in your organism. How many ribosomes are there? 20,000? 30,000? Now you've got 20 or 30,000 times as much fluorescence in that cell than if you target a specific gene. Furthermore, if you target a specific gene, each organism will show up as a single spot, right? Whereas if you target the ribosome, the whole cytoplasm glows. So there's a huge difference in what you get. Now, people use fluorescent in situ hybridization in eukaryotes all the time, and in which case they're targeting the location of specific genes in a chromosome or specific messenger RNAs or that sort of a thing. But you're looking at cells that are enormous. Here we're looking at bacteria, and so ribosomal RNA is perfect. There's an interesting technique, though, that you can use to improve that. Instead of just looking at a static slide and looking for a cell that either glows or doesn't glow, there's a better way to use this technology that uses confocal microscopy, which allows you to get higher resolution than a typical fluorescent microscope. It allows you to collect images in color, and it allows you to collect images in 3 dimensions. So instead of taking a 2-dimensional slide, you end up with a 3-dimensional reconstruction. Now you guys do this a little bit in lab all the time, right? You look through the microscope, you're at high power, and you focus up and down to get different things in focus. You can do this with a fluorescent microscope. Imagine capturing, moving that fine adjustment, capture an image, move it a little bit, capture another image, move it a little bit, capture another one, and you could stack those up and make a 3-dimensional image, right? That's basically what laser confocal microscopy does. Let's take a look at this video. This is just one I happened to pull up on YouTube.
Confocal microscope is used to overcome these obstacles. To capture an image, a beam of light illuminates the specimen. Mirrors then focus the reflected light towards a pinhole-size opening in front of the camera. Only the light that is from a single thin, in-focus slice, the focal plane, passes through the pinhole.
So the trick here is to increase resolution. So the laser is here, you've got a sample here, and you're going to sample in 3 different slices. The laser hits the point right here, it's focused on this point. Now, in order to get to that point, it's got to go through these other layers first, right? And so there's going to be some light scattering from that. But light that's scattered will be coming in a different direction. And since you're collecting data through a pinhole, as it bounces off this mirror, it's going to miss that pinhole. The result of this is that it allows you to capture only images from that plane. And all the fuzz that comes from above and below it is removed. This increases resolution dramatically. This image, it looks a little more complicated than it should. And the reason is because the way these things are captured is that you're using a laser rather than white light. And so instead of capturing the whole image at once, you use the laser to scan the image, just like an old-fashioned TV tube. And so if this were the microscopic image right here, instead of seeing the whole image at once, the laser scans back and forth down the whole thing, and it captures one pixel at a time. Now I can do this very quickly, of course. But that's what all this stuff is supposed to mean. And so you focus in on one layer, you scan it to capture the image, and then you shift the focus up or down just a little bit, you capture another layer, and you repeat this process over and over again.
Sounds like 3D printing.
3D what?
Sounds like 3D.
It is like 3D printing, only you're doing the reverse.
By
You're
blocking
capturing
the
image
out of
instead
focus or
creating
blurry
the
light from the specimen, the confocal microscope is capable of creating a much sharper image than a wide field
because of
microscope.
the fuzz of looking
A
at different
technique
layers.
often combined with confocal microscopy is fluorescent imaging. Fluorescent dye molecules are attached to specific parts of a sample so that only those parts are the ones seen.
So this is another key. Instead of just looking at an object like you would under phase contrast microscopy, this is the fluorescently labeled object. And so the image you're collecting is you excite with a laser of a particular wavelength, or typically with a filter. And then what you're capturing is the fluorescence that it emits at a different wavelength.
Microscope. Dyes are constructed for molecules like proteins and nucleic acids, or to
Ribosomal
localize
RNAs.
a specific structural region like the mitochondria or nucleus. More than one type of dye can be used on a specimen, and by changing the wavelength of light, different parts of a sample are able to be distinguished. The confocal microscope also allows for the construction of 3D images of specimens. Images are captured at multiple planes, ranging in number from 2 to 200, to create a stack of images. Once all the slices are stored in the computer, the image data can be combined computationally to construct a 3D image. As stated earlier, confocal microscopes have the ability to image live specimens. Images are taken over.
So this is a difference. In this case, they're not soaking in a phylogenetic probe. They're staining things with things like fluorescent antibodies or something where they can do this in living cells. That's not what we're talking about today. But this then is a little video, a two-dimensional image. Very, very often, although she doesn't mention it, when you capture these three-dimensional images, since all of the slices are in focus, you can collapse them all into a single layer and get a very high-resolution two-dimensional image of your object. Basically taking Photoshop and just combining all the layers in one.
Period of time at regular intervals and are combined to make a stunning time-lapse video.
So that's laser confocal fluorescent microscopy. I'm going to save this video real quick.